Okay, I think we'll begin. I want to welcome you all to the second part of the BERD clinical trial series. This is an introduction to clinical trials, and what you definitely need to know is something about our presenter because we have a very uh, excellent presenter and Dr. Austin Miller. He's in the Department of Biostatistics and Bioinformatics at Roswell Park, Assistant Professor of Oncology, and he is also in the UBC TSI Core Operations for BERD. And I'm going to turn it over to him. He has quite a lot of information to give you. And what we're going to do is he has some slides that you can stop and ask questions, but we also want you to be able to organically ask questions as well. So if you have any questions, feel free to ask them, type them in the chat, and I will wait till the end of the slide that he is presently on in order to ask him your questions as well. So please feel free to interact as much as you need or want to, and I'm going to turn it over to Dr. Miller. Thank you.
Great, thank you. So I also understand this is like two 45-minute chunks, so I will set myself a timer so that I know exactly what I'm going to ignore. Right. So this is the introduction to clinical trials. So a little bit about me. So I got my PhD in biostatistics in 2009. I've been working in clinical trials since 2006. I started as an intern at Roswell Park. Currently, I'm the director of biostatistics and statistical programming in the Clinical Trials Development Division at the Roswell Park Department of Informatics, Biostatistics and Bioinformatics. As you know, the longer someone's title is, the less meaningful it might be. So I am also, as part of that, the Associate Head of Biostatistics in the NRG Oncology Statistics and Data Management Center, the Buffalo office. And that makes me the lead statistician for GYN clinical trials conducted through the NRG cooperative group. And I'll talk about the cooperative group in just a minute. So we work on all the phases, right? Phase 1, phase 2, phase 3. We spend most of our time in phase 2 and phase 3 trials, and occasionally we get a definitive FDA regulatory intent study, which is all very exciting. And we can certainly talk about all that if you like. So a little bit about the National Clinical Trials Network, the NCTN. So this is a NIH-sponsored network. There are 5 groups within that network. Hold on, pointer, laser. There's the Southwest Oncology Group, Children's Oncology Group, ECOG Akron, NRG, and the Alliance. And these are sort of centralized all around at different points all around the country. Each group, like for example, the NRG Oncology Group here, has a bunch of member sites. These are hospitals that are participating in our clinical trial. Each group has a tissue bank, so tissue specimens that are collected in the course of the clinical trial get sent to the tissue bank, so they're all centrally located and available for ancillary studies and other sorts of investigations. There's a statistics data and management center for each group, and there's an operations center for each group. So the NRG Oncology in particular has about 1,000 member sites, member hospitals, and upwards of 2,500 participating physicians. So it's a pretty good size group. A little bit of this, so I'm just going to share this. This is sort of the context that I'm coming from. So the context that you're in might be somewhat similar or completely different. I think the concepts are the same, but I think it's helpful for you guys to understand where I'm coming from. And so if you hear, I'm going to take some vocabulary for granted and if that vocabulary doesn't make sense to you, please stop me so we can square all that away. So in the drug development cycle, there's a drug company that has a super fantastic drug that they absolutely need to get to their patients in the market. And in order for them to get to the market, they have to go through the FDA, right? And in order to get through the FDA, you have to have a well-developed, statistically sound, well-controlled, standardized data collection, systematic follow-up, reproducible clinical trial, right? So these are the sort of general characteristics of what the FDA is looking for. The challenge here is that the drug company doesn't have access to patients, and the FDA does not have access to patients, and so somehow those patients have to magically appear in that clinical trial, and that's where the NCTN comes in. So we partner with the drug company on behalf of the NCI, or on behalf of the NCI, and we work with the drug company to sort of define the protocol, the patient characteristics, the eligibility criteria, all that sort of stuff. And we sign up hospitals by the hundreds in order to gather up patients by the hundreds in order to populate that clinical trial. There are other ways for the drug company to go. They could go to a CRO, a contract research organization. In my group, we also work with the GOG Foundation, which is technically a CRO. And then this group forms a team to conduct either one or two or three multiple clinical trials that we would then, if they show efficacy, then we would send that to the FDA along with in company with all of these other collaborators. Then the FDA approves it, and it goes to market. Each one of these protocols is no matter how small the trial is, they are a monumental effort. They take many sites, many physicians, many patients, costs big money, right? The average cost for a patient in an NCTN clinical trial can run upwards of $50,000 to $75,000 per patient. And because there's so much money involved, there are many stakeholders. And so there are lots of revisions to the protocols, et cetera, et cetera. It becomes rather complicated. From a development standpoint, every trial starts off as a concept. It goes through multiple committees, revisions. There are many false starts. We only get probably 2 out of 10, maybe 3 out of 10 concepts actually make it through to the clinical trial phase and actually enrolling a patient. So there are lots of layers of science that have to be reviewed and agreed to by pretty smart people in multiple committees. After about a year in the concept phase, we move on to developing a protocol and a statistical analysis plan. This part takes about 6 months. After that, we have to get into database building, data collection, monitoring, reporting, fixing all the data, trying to correct the things that goes wrong because nothing ever goes as planned. And then at the end, there's an analysis, primary and secondary endpoints, translational research, quality of life, ancillary projects, lots of manuscript developed that happens at the end. All right. So I think Dr. Lamont showed something similar to this in his presentation. This is the FDA's layout for a clinical trial progress. This is a little bit different presentation than you saw before. So phase 0, these are exploratory IND studies. They generally have no therapeutic intent. They're using subtherapeutic doses, and it's a very short exposure. So these, in our setting, this often happens, and we call them window of opportunity trials. So a subject or a patient enrolls. We collect a tissue sample of some kind. They get some sort of quick 7 to 10 day exposure to a drug, and then we take another tissue sample, and we're looking for the effects of the drug on that tissue sample. So these are very small trials, maybe on the order of 10 to 15, maybe 20 patients, and they go very fast. We don't do a lot of these. Phase 1, we do more of. These are dose-finding studies. You're looking for the safety profile for the drug. Maybe we're testing out different delivery methods. Maybe you get some preliminary efficacy result, but generally the number of patients is rather small. So the efficacy signal might be on the weak side. In particular, there's no control arm in a phase one, typically. The primary endpoint is dose-limiting toxicities, anywhere from 15 to 50 odd patients, although we did have one GOG 9929 that had upwards of 450 patients in it, multiple arms. About 70% of phase one studies result in a phase two study. So phase two and phase three is where we spend most of our time. Phase two is this is a preliminary test of efficacy, and we're going to monitor for side effects. You could have historical controls, which is less common. Randomized controls is more common. The common primary endpoints are sort of objective response rate, maybe progression-free survival. Sample sizes range between 330 and up to 100. And about a third of phase two trials go to a phase three setting. So in phase three, we're looking for a definitive test for FDA approval. So this is a direct comparison of efficacy of a new treatment to a standard of care. Always has randomized controls. The primary endpoints are progression-free survival, maybe overall survival. There's at least 200 patients on these, up into the 500 and 600s, maybe per arm. About 25% of phase three trials make it into phase four, which is a monitoring phase. So if you just multiply all of these percentages at the bottom, it turns out that somewhere around 5% of phase one studies, the drugs in phase one studies make it to the market. Right, so just a little overview there, right? Dr. Lamont also talked about levels of evidence, or the, sort of an evidence-based medicine paradigm for thinking about the value of information or the quality of information that's in a clinical trial. So if you look on the bottom here, we've got unpublished sources, expert opinions and editorials, case series and case report studies. These are sort of individual patient studies, or maybe very small cohorts. Non-comparative trials, when you start getting into uncontrolled cohort studies or single-arm trials, maybe some of you've heard of a single-arm Simon phase two. This is often a design that's used in a non-comparative study. Then moving up the trail a little bit, you get into controlled cohort and randomized controlled studies. This is where your levels of evidence start to increase quite a bit. And in the phase three range, it's the randomized clinical trials that provide the most solid evidence, I think, that we can get. There are a few, once in a while, there are meta-analysis and systemic reviews that are done that are also very compelling, but these are, I think, more unusual. So getting a drug into the market is in this randomized controlled phase three trial area, if you will. Okay, any questions about any of that?
There are no questions in the chat at this time.
Nothing in the chat. Let me close that then. All right. So just a real quick, this is some things that I believe that you have covered, that have been covered previously. So the assumption here is that the attenders have little or no experience developing research studies, and I'm going to call that clinical trials instead of research studies. Dr. Lamont talked about some randomized trial concepts, the broad categories of prevention, efficacy, and effectiveness, some of the components of a clinical trial, the research question, the population, how we allocate treatments, maybe some outcomes and some monitoring. There were a lot of examples, but not I think, a lot of specifics. He also talked about some of the design components that goes into a clinical trial, specifically randomization, stratification, internal and external validity, etc. He did ask me to kind of hit randomization stratification again and to focus on the intent to treat population analysis paradigm. And so I made sure to include that here on his behalf. Okay, these are our learning objectives. Part one, we're supposed to understand the importance of a well-defined primary and secondary outcome in clinical trials. differentiate between continuous and categorical outcomes and their implications for analysis. And then in part 2, grasp the basic statistical concepts underlying a clinical trial analysis and recognize the appropriate tests for different types of outcomes and designs. So I think I'm going to run out of time right about in this last space, so I don't have a lot of information on that for you today. So let's talk about randomization. So in order to talk about randomization, I have to talk about selection bias. This goes all the way back to 1904, and Carl Pearson, in his publication, he talks about the problem of selection bias. What he writes in his paper is the thing that we should do, right? He's worried about an inoculation, essentially a vaccine trial, right? And his plan for adding patients onto a vaccine trial is to keep a register of all men who volunteered only to inoculate every second volunteer. So he's got picture a line of men standing up outside of his hospital waiting to get a vaccine or not. And so the first one that comes in, he gives a vaccine to, the second one he doesn't, third one he does, the fourth one he doesn't. It's just sort of this every other one decision rule, right? And in his world, or in his view, in this way, any spurious effect really resulting from a correlation between immunity and caution would be gotten rid of. So this idea of immunity is something innate about the patient that they're bringing to the trial, and caution is an attitude about the patient relative to whether or not they should get a vaccine or not, right? So this is the first, not randomized, but it is a way of enrolling patients onto a study. And in his paradigm, what that sort of on and off or every other patient enrollment strategy does is it eliminates any spurious correlation that may be produced between the attack or the absence of an inoculation, right? So in theory, the patient doesn't know that, it shouldn't know whether they are an even number or an odd number in the line. And this is the problem with Carl Pearson's original thought, but this is where the idea comes from. So let's talk about selection bias quickly. So if you're a drug company, for example, you have some new whiz-bang experimental treatment that you want to take to the market. And your primary outcome in this case, just say, is time to death. And so we want to see if our experimental treatment improves the survival of patients that receive it. In order to do this effectively, we need to have a reference arm. And that reference arm is probably some standard of care that the patients are getting right now. The experimental arm might be in addition to the standard of care. It might be a wholly different treatment regimen. It's not important at this point. Only to say that if you're going to have an experimental arm, you need to have a comparator in order to make any interpretable findings. And then you need physicians, right? And so you go to the cooperative group and you say, help me enroll doctors who have access to patients that meet my eligibility criteria. But those physicians, they come to the study with opinions about the treatment, maybe limitations or access to the treatment, some experience with the treatment, or maybe they've read something about it, maybe a phase 2 or some earlier findings on that particular treatment. They have opinions about the cost-benefit analysis, how the patients will benefit, et cetera, et cetera. And the point here is that they come to the trial with a lot of maybe information, maybe opinions, maybe guesses about what the treatment is expected to do, whether it will be efficacious, how toxic it'll be, how expensive it'll be, et cetera. But they come with all of these opinions. The physicians then enroll patients, and it turns out those patients have a similar set of biases when they come in. Their biases related to socioeconomic status, Some patients have more or less access to care. There are different disease and health factors that they bring. They have questions about the cost benefit analysis also, and so they have opinions about how or when or what to expect from this treatment. All of these exogenous factors have a potentially biasing effect on whether the physicians would enroll a patient into the experimental arm or not, whether the patient wants to be in the reference arm or in the experimental arm or not, right? And all of these exogenous factors over here prior to the randomization line potentially corrupt the assessment of the experimental arm relative to the reference arm against an outcome of time to death. So for example, if a physician doesn't think that the treatment arm works in a specific population, then he might not add those patients to that treatment arm. And if there are significant prognostic differences in those patients that he selects, then it introduces biases in the treatment effect estimates. Right? So what we want to do is break the effect of these exogenous factors on the treatment assignment. Right? And that's what randomization does. Is it takes all of these opinions and all of these patient level factors, and it balances them out between the experimental arm and the reference arm. So in particular, anything that is prognostic gets balanced out if, of course, the sample size is large enough, and the randomization holds, right? So this balances not only the factors that we have collected or analyzed, it balances out things that we haven't measured also. And so this is the way that we get a really clean estimate of the treatment effect of the experimental arm versus the reference. So random allocation actually means that each patient has a known chance, usually an equal chance of being given each treatment, right? In other words, when the patient comes into the clinic, they cannot predict what treatment they will get, right? So technically, there's no such thing as a randomized trial. There are randomized treatments, but patients are not randomized. We're assigning treatments to patients and not the other way around. But this is the basics of a random allocation is that the treatment that the patient will get is not predictable in advance by the patient anyway. This is different from concealment, right? So allocation concealment, sorry, not different from the, so allocation concealment, this is how we prevent selection biases, right? This protects the treatment assignment sequence before and until the allocation of the treatment, the randomized treatment. You can always do this, right? This is a simple and easy thing to do. We can make it complicated if we want, but it doesn't need to be. Blinding is different. Blinding protects the sequence or the treatment allocation or treatment assignment after it's been allocated. So we're preventing assessment biases, collecting data in a way that favors one group or a treatment group, so blinding protects against that. Blinding cannot always be implemented. Sometimes it's hard. You can imagine a surgical trial, right? So if patients are going into surgery, they know what surgery they're getting, and the physician knows what surgery they're going to give, right? So a good example of this is we're working on a HIPEC trial now, where the patients are randomized after they get a cytoreductive surgery, and before they get the HIPEC add-on to that. So the randomization happens while the patient is unconscious and on the table. The physician gets an envelope at the end of the cytoreductive surgery, opens up the envelope and says, okay, we're going to give this patient a HIPEC treatment or not. Reasons to randomize, there are many. There's a whole raft of references on this. I've given you 4 below in the slide below here, so you can take a look at those if you like. Reasons to randomize are to ensure the similarity in the treatment groups on both known and unknown confounders. Importantly, We're going to balance prognostic factors, maybe through stratification, which I'll talk about in a minute. This is at least asymptotically balanced if you have a large enough sample size. If you don't have a large enough sample size, then maybe some stratification will help here. This is at the very heart of convincing an audience that your findings are true. So if you're trying to change the standard of care, or you're trying to make people believe that your experimental arm was effective, then you have to have some way of convincing them that there is no confounding effects in your treatment estimates. Randomization is the most compelling way to establish that an intervention causes the clinical outcome. This is a causal analysis because we randomized. If you don't randomize, you can't claim that directly, right? There's also some benefits to randomization in that as the sample size increases. So if you only have 10 or 15 patients and you're going to randomize half and half to the different arms, it's less effective. The randomization is less effective than if you have 50 or 60 or 100 patients. So again, the randomization itself, all by itself, provides the basis for inference. So there's greater clarity in interpreting the results. I get more precise estimates of the effect size and the sample size requirements if we randomize. Prognostic factors get balanced across treatment groups if we randomize, and the statistical significance tests at the end of your studies are considered valid. In the absence of randomization, there's confounding, and the test may have bias in it also. Some simple ways to randomize. So, I mean, you could do the every other patient patient allocation rule if you can keep the patients from knowing what their treatment assignment will be. Otherwise, we use randomization. A typically easy way to do this is by permuted blocks. Here's a block size of 4. If you look in here, block 1, the first patient gets randomized to arm A, the second patient to arm B, the second patient to arm A, right? The third patient to arm A, the third to arm B, etc. In the next block, it's different. It goes B, B, then A, A. Then the third block is a little bit different, B, A, B, A. This is the standard way that we do randomization within these blocks. So this ensures the treatment balance throughout the trial, and in this case, I'm thinking about that first interim analysis where you've only got maybe a third to a half of the number of patients that you expected to accrue, and so we like to ensure that balance at an early point in the study. If you have a large enough study, you don't really need to do permuted blocks or anything like that. As long as you have randomization, in the long run, all of those balancing properties should work themselves out. This protects against selection bias if the concealment works. So if the patient does not know what drug they're going to or what arm they're going to be on at the time of randomization. The downside here is the pattern might be predictable. So if you only have a couple of let's say, doctors enrolling patients, then the next treatment they might be able to figure out what the next treatment is. In a cooperative group, we have so many doctors and so many hospitals enrolling at the same sort of simultaneously that they can't figure out. There's no way that they could figure out what that allocation is. So something to be cognizant of if you're going to use permuted blocks. Any questions?
There is nothing in the chat at this time.
A quiet group. All right. All right, stratification. So this is the schema from the combo match N4 study. It's actually half the schema. There's my pointer. There we go. So over here, cohort 1, this is an ovarian cancer cohort. There is a cohort 2, which is endometrial cancer, and I've left that off because it's virtually identical to this one. So patients come in, to the ovarian cancer cohort on N4, and they are stratified based on whether they have low-grade serous ovarian cancer or some other part or serous ovarian cancer, right? So we stratify on the prognostic factor, which is whether it's low-grade serous or other. And then within this group, right, within this stratification factor, the patients get randomized either to the experimental arm, which is selumetinib plus olaparib, or the control arm, which is selumetinib alone. What's not shown here is that these patients on the reference arm, after they have disease progression on treatment, they can cross over to the combination arm, right? So the point here is that randomization happens within the stratification levels. So why stratify? So stratification balances the treatment allocations within prognostically significant subgroups. This particularly helps with smaller trials of something less than 100 patients, say. It's possibly important at an interim analysis where the sample sizes are small. And this is really the main reason that we do this. Other benefits, you get better estimation of a treatment effects within subgroups that you stratified on. There's possibly some reductions in the type 1, and type 2 error rates. There's some estimate says that by stratifying on important prognostic factors, it's comparable to adding about 14 patients to your trial, right? So you get a little bit more efficiency out of it. Unfortunately for me, the maximum desirable number of stratification factors or levels is not known. And so the clinicians think that there's lots of prognostically significant factors to stratify on, and they want many. And unfortunately, if having too many can lead to some problems. So we recommend for sort of keeping that number small, maybe around 2 to 4 at the most. So why is fewer better? Because when you have fewer stratification levels, that you get more patients in each stratum, all else being equal, right? So this simplifies the randomization and trial administration. It improves statistical efficacy. So it's more likely that you'll get equal numbers of patients that are assigned to each treatment. If you have too many, then you end up with strata that have no randomization blocks completed, right? So this is over-stratified. You have empty cells in your analysis, right? This causes imbalances in the prognostic factors, especially when you're using permuted block randomization scheme. So if you're going to use permuted block, keep the number of strata in your randomization small. There are several references here that might be helpful. Some rules of thumb. Just remember that the number of strata is the product of the number of levels in each strata. So if I have 4 stratification factors, and each one of those has 4 levels each, then I have 16 blocks, right? 16 cells from which to randomize patients that are coming in, right? So if with this many strata, if you have a small number of patients, then it gets hard to, you can't ensure that you have patients in every cell, right? So statistical methods then estimate the treatment effect within each strata and aggregate the results. So if you don't have patients in a strata, that gets somehow merged together in the aggregate results. And this can be problematic when you have empty cells. So the number of patients that you have in each cell, of course, depends on the number of patients that you're going to enroll, the expected number in each strata, and the strength of the prognostic factors. So the main reason for stratifying in maybe a larger trial is at that first interim analysis, right? So what we want to do at that first interim analysis is get a reasonable estimate for the efficacy of the experimental arm, right? And to do that, I have it with a relatively small number of patients. I have to have enough patients in those cells to make the interim analysis estimates be good. Any questions about that? Could you please elaborate on in what way stratification possibly reduces type 1 and type 2 errors? So stratification, you want to stratify on prognostically important factors, right? And because, here we go. Let me back up. So let me go here, right? So if we didn't have this, stratification layer, right, then patients would come in here and get randomized to one of these two treatment arms, regardless of what that prognostic factor was. And so when you have a small number of patients, right, you may end up with an imbalance in that prognostic factor in, maybe the patients with better prognosis end up disproportionately in the experimental arm, which would make your experimental arm look naturally better, right? So it happens when you have small sample sizes, which for us generally happens at an interim analysis. So if you control the randomization within each one of these prognostically important stratification factors, that stratification factor, that prognostic difference is automatically balanced out in the randomization process. Does that make sense? Tough to have a two-way conversation.
She said, Young Six said, I see what you are saying. Thanks.
Got it. Thank you. All right, so let's talk about the intent to treat principle, and I think I have 8 minutes to do this. This is at the request of Dr. Lamont. I guess there were some questions about this in his lecture, which I'm not aware of what those questions were. So if I'll blow through this, I guess fairly quickly, and I'm happy to pick up any questions at the end. So the intention to treat principle goes back a very long way, and there's some of these references below, particularly that McCoy reference, 2017, which is very easy to read and explains things pretty well. So you're happy to take a look at that. And some of what I got here was pulled from there, but I also like this Lakin 2000 paper that's in controlled clinical trials. Is very helpful. This has been very helpful to me in some other projects that we've done. So the intention to treat principle is a complete trial strategy for design, conduct, and analysis of the study, right? The mantra is once randomized, always analyzed. So the intention to treat analysis includes all the patients, all the events, all the outcomes, right? We're going to analyze patients by the treatment assigned at randomization, regardless of their compliance, protocol deviations, withdrawal, or anything else that happens after randomization, right? So we're going to randomize everyone that we're going to analyze everyone that we enrolled, and we're going to do it within the groups, that were defined at randomization. And what this does is it preserves the balance of the prognostic factors. It preserves the number of patients, the events. It makes everything sort of add up. And this is the preferred way to do it. The intent to treat treatment effect estimate is generally conservative. So it includes non-compliance, protocol deviations, which represent real practice. Excluding these patients may artificially inflate the treatment effect estimates, right? So if you're doing a superiority analysis in particular, the intent to treat principle gives you a conservative estimate of how effective your experimental arm is, right? This preserves the integrity of the randomization during implementation and analysis beyond just the randomization, right? Preserves the sample size and number of events. By doing this, I maintain the statistical design elements, namely the type 1 and type 2 errors. This limits inferences based on ad hoc subgroups. So which is why we say we shouldn't just leave out patients who were non-compliant with their treatment. Let's not leave out patients who withdrew consent unless we have to. Those sorts of things, right? This makes your estimates more generalizable because it's more reflective of what happens in the real world. The FDA guidelines, they don't say that it has to be the primary analysis. It often is for us. It's usually the intent to treat analysis that we would call primary. The FDA just says that in your submissions, you need to include an ITT analysis using all the randomized patients. So it doesn't have to be primary for them, but it does have to be there. This is even if the preferred analysis is based on some reduced subset of the patients with data, right? So the mantra here is once randomized, always analyzed, and it is largely the preferred reporting outcome, the preferred primary outcome. Okay, so was there something about that in Dr. Lamont's talk that was not clear that I can clarify?
If anyone has any questions about that, please feel free to ask at this time. I think you offered clarification. Thank you.
All right. All right. So this is an example of a study that we published in 2022. It's GOG 281. And I hope this will just give some sort of a real world example for how these things, how the stat section in your protocol is set up, I think. So here is the schema for GOG 281. This is a randomized pointer. This is a randomized phase 2-3 study to assess the efficacy of trametinib in patients with recurrent or progressive low-grade serous ovarian cancer or peritoneal cancer. We called it the LOG study. We enrolled 250 patients. The primary endpoint was progression-free survival and had several secondary endpoints there. So we randomized patients 1-to-1 to a control arm versus an experimental arm. The control arm was investigator's choice. They could use letrozole, tamoxifen, paclitaxel, pegylated liposomal doxorubicin, or topotecan. These are the drugs that are currently used to treat low-grade serous ovarian cancer, or were at the time. In the experimental arm, the experimental arm was made up of trametinib only, right? So we were comparing trametinib to whatever the standard of care was that the investigators got to choose. Importantly, on the control arm here, the way the control arm worked is this was a stratification factor. So before the patient was randomized, the investigator had to declare what drug they would give to the patient if that patient was randomized to the control arm. And then once that decision was made, there was a randomization, and the patient went to either the control arm or the experimental arm. Obviously, if they went to the experimental arm, they got trametinib. If they went to the control arm, they got whatever that drug that the physician declared up front. So this became a stratification factor. We followed those patients to disease progression, or it was a progression-free survival endpoint, which we'll talk about a little bit later. Patients on the control arm, once they progressed, were allowed to cross over to the trametinib arm, again, after progression. And you can see how that might have obscured some of our overall survival results, but I don't think I may not get into that too much. So this is GOG 281. This was a practice-changing study, right? So you can see here, the median time to progression on the trametinib arm was 13 months compared to 7 months in the standard of care. The hazard ratio was 0.48 with a p-value of less than 0.001. So this is a very strong finding in favor of trametinib. And this is now in the NCCN guidelines as the treatment of choice for low-grade serous ovarian cancer. So if you read this, just for example, so the median time to progression, here's the line where 50% of the patients would have progressed. You can see on the standard of care arm, the time to progression is a lot shorter than it was on the experimental arm, on average about 5 months shorter. So a strong result. A strong result following a 10-year timeline. So we started concept development in February of 2012. Accrual finally opened in February of 2014, 2 years later. The interim analysis for futility happened 3 years after that in 2017. The primary endpoint matured 2 years after that in July of 2019, and we finally got it published in September of 2021. Right, so that sets us up for a discussion of statistical endpoints. So the FDA has several guidance documents on endpoints that have come out sort of sporadically over the years. The first one to introduce standardized PFS, progression-free survival censoring rules, came out in 2015. In 2018, they expanded on that, sort of modified their definitions a little bit. And then in 2021, they sort of recalibrated their thinking altogether, and in a way that harmonized with the European Medical Agency's rules. And so to provide as much sort of cross-Atlantic collaboration as we could get. So it really helps to have the Europeans accruing to our studies. It makes everything go a bit faster. So the primary estimand for GOG 281 was progression-free survival. And I put this up in all of its gory detail to say that when you're designing your study, that you should put the same amount of thought into whatever your primary endpoint is. So this framework is pretty clearly laid out in the FDA guidance document from 2021. This addendum of estimands and sensitivity analysis in clinical trials. You can get that off the FDA website whenever you like. But they have these they have these 4 attributes of an estimand that you need to fill in. So the first one is your population. You need to describe what that population is. For 281, it was women with recurrent low-grade ovarian serous carcinoma of the ovary or the peritoneum, and they were previously treated with some platinum-based chemotherapy. So they'd already progressed on something else. The variable or the endpoint here, in my case, this is progression-free survival or PFS. It's defined as the time from randomization to progressive disease per RECIST version 1.1, which we'll talk about a little bit later. So time to progression or death, whichever comes first. Intercurrent events. So these are other events that could be observed that might compete with or maybe conflict with my primary endpoint of progression, disease, or death, whichever comes first. So these are, you can start non-protocol therapy or anti-cancer therapy. This is a hypothetical strategy in the FDA's parlance. And what I'm going to do with these patients at the final analysis is I'm going to censor them I'm going to censor their progression-free survival time on the date of the last scan prior to non-protocol therapy. So this comes out of an FDA recommendation published, I think, in 2018. Other intercurrent events, the patient could develop a new primary disease. We're going to call this a treatment policy. We're going to censor, I think we censored them. I think I got this wrong in this slide. So I think we censored them. A new primary disease means that they had some other disease that came up that was not the recurrent low-grade serous carcinoma. They had, I don't know, like a brain cancer or a lung cancer or something like that. And so what we normally do is we would censor them. Patients who withdraw consent, this is a hypothetical policy again. We're going to censor these patients on the date of the last scan before non-protocol therapy. There's some patients that got no treatment at all. right? So this again is hypothetical. We're going to censor them on the day after randomization. So what this does for me is it allows me to include all of the patients in the study, whether or not they were treated, right? So this is pretty clearly a way to handle or to facilitate an intent to treat analysis down road. And then there's the population level summary is the last attribute. Um, this is the how are we going to tell the world how the study did. So in this case, my primary analysis or my primary treatment effect estimate was the progression-free survival hazard ratio for trametinib versus standard of care therapy, estimated by a Cox regression model stratified by the factors declared at randomization. We use using an intent to treat principle. Patients will be analyzed in the treatment group assigned at randomization, regardless of compliance with that treatment. Right? So my point here is that this is the primary endpoint. It's not selected easily or sort of frivolously. It's not frivolous at all. And if you're going to put this in your trial as your primary endpoint, you should think about all of these different aspects of that endpoint, so that you are very clear, the investigators are very clear, and perhaps more importantly, the people who read your manuscript are very clear on how you are assessing the treatment effect of your experimental arm. So the interesting or perhaps new component of this in the newer FDA guidance is this intercurrent events block. So we always assumed these things, and they have other guidance documents that detail these things, but they never called them out like this before. They sort of had recommendations in their guidance documents, but they never called them out so explicitly in the definition of the estimate. And the issue here is that for these intercurrent events, is it gives you a chance to really think through them. So on a time to event analysis, there are lots of things that can happen to the patient before she gets to the point of progression, right? So let me hide this thing. So consider patient number one, right? She comes in, we follow her for a time, and then she discontinued treatment because it's really not working for her. So she didn't progress per se, so she didn't meet the endpoint policy. She met the endpoint somewhere out here, right? Toward the end of her, at the end of her treatment cycle, but she discontinued treatment back here. So you can see that this might be something that would influence the primary estimate, right? The primary treatment effect estimate. So if this patient, for example, is on the experimental arm and she discontinues treatment for lack of efficacy, she just stops taking the treatment, then what happens is your experimental arm looks ever so much more like maybe your control arm if she comes off any kind of treatment altogether. So in this case, what we did with these patients is we said it doesn't matter that she discontinued treatment for lack of efficacy. It doesn't matter that she discontinued treatment at all. We're going to follow her until we get to the progression endpoint, right? So patient number two, you follow her along here along this dotted line. You see nothing really happens to her. She gets out here, treatment gets completed. Treatment complete in the case of GOG 281 means that she had disease progression, right? And so this is actually the perfect scenario for GOG 281. Patient number three, you follow her along, and she discontinued the study, right? So she came off altogether. So she doesn't want to be treated anymore. We're not going to do any more scans. We're not going to have any more follow-up. And so the rule is that we're going to censor her on the date of the discontinuation, and the logic just sort of keeps going on. Rescue medication in the case of patient number four. This means that she got something because the treatment, the assigned treatment, really wasn't working for her, and so there were some issues here. And so we might censor her here if you thought that this was an indicator of progression that wasn't picked up on a scan, for example. And so these sorts of scenarios are helpful in figuring out how you're going to define your primary endpoint. These intercurrent events often cause missing information, right? And so the real question on the FDA's in the FDA's formulation is, what are you going to do when patients don't meet the strict definition of your primary endpoint, right? And all that needs to be very clearly laid out. Okay, so I talked in detail about progression-free survival endpoints. Oops. So here's a couple other endpoints. Let me get my pointer back. Overall survival. This is the time to randomization, from randomization until death from any cause. This is, in many ways, a preferred endpoint because it is precise and easy to measure. We know when the patient died. There's really no question about whether they're alive or dead. The date we know exactly, and so we can calculate everything as precise as any endpoint that we have, right? Bias is really not a factor in the endpoint measurement, right? Because it's just a date. It's a fact and everybody knows it. There's no questions. It's hard to assess if patients switch treatments, perhaps multiple times, and this is one of the things that makes it a little bit difficult. If your patient prognosis in terms of overall survival is fairly long, then there's a good chance that they're going to switch treatments on you several times, and so you have to figure out how to deal with that, right? So this is the difficulty: is a long follow-up period. Subsequent cancer therapy potentially confounds the survival analysis endpoints. So we like this one because it's very easy to measure, and it is the most important endpoint from a patient perspective. Unfortunately, with long follow-up periods, it can be hard to assess in terms of a treatment effect. So if we can't use overall survival because, for example, the follow-up period is too long and it makes the study infeasible, then we have to find some other clinical endpoint that happens faster, right? And so this gets into endpoints that are based on tumor assessments, right? In which there are several problems with these. So tumor assessments come from radiologic tumor assessments or CT scans, right? So these directly measure components of the disease. In this case, they measure the size of certain tumors, right? And this is good because those size changes commonly trigger treatment effects or treatment decisions in clinical practice. So if you have a disease that's tumor-based, and you can measure those diseases, and physicians do this. If there's a 20 or 30% increase in a tumor, then they'll say the current treatment is not working. We need to move on to something else. So it's the basis of some decisions in clinical practice. And this is both good and bad from the standpoint of a clinical endpoint. So CT scans is the tumor assessment. So in your world, in your practice, you might have other ways of measuring some disease-related component, and those could be used as well as long as they're well defined. So from using a CT scan, measuring from a CT scan, you introduce the potential for bias and uncertainty, right? So several types of bias: investigator bias, assessment time bias, follow-up time bias, and toxicity bias, right? Investigator bias is the investigator has an opinion about one of the arms on your drug, say it's the experimental arm, and so they may tend to overstate or understate the measurements of their tumors, right? Assessment time bias is there's a systematic difference in the timing of the scans. So for our studies, we actually create a scan scheduler. It's just a spreadsheet that tells every patient when their next scan is going to be. And it's not tied to a visit schedule per se. It's not tied to anything other than a calendar point, right? So this makes it a little inconvenient for patients, and so we give them a 14-day plus or minus window. But the idea is to prevent assessment time biases by having all of those tumors evaluated at a constant interval. If they get off the interval, we say they need to get back on the interval as soon as medically feasible, right? And that's all about assessment time biases. So you can imagine if the scan interval on the experimental arm was longer than the scan interval on the control arm. So in that case, the patient might progress at the same time on either arm, but it's measured later on the experimental arm than it was on the control arm, and that makes the progression time on the experimental arm look artificially longer, for example, right? So assessment time biases, follow-up times, if we're not following patients as long or under the same rules, different toxicities may influence whether patients get scans or not. And so there's lots of complicating factors in endpoints that are based on tumor assessments. Because of that, the FDA is a little bit skeptical. And so they may require confirmatory evidence from a second trial. So if they really don't like the way these biases were controlled in the first trial, they may ask for a second one, which would be rather expensive. Accuracy in measuring tumors can be different based on the tumor setting, where the tumor is, how easy it is to measure, right? You have to be able to see it and measure it the same dimension each time. So this can get challenging. So the measurements may be imprecise. Sometimes they're using calipers on a scan image to see, so to get a dimension, and this is imprecise. Generally, what the FDA wants is reviewers that are blinded to the study treatments. So we call this a BICR, or a blinded independent review. This is actually often conducted after this trial is over, and if there is a treatment effect on the primary endpoint. And if in that case, and if it's a good, strong treatment effect, then the company will hire central blinded independent reviewers to look at all the scans and confirm the tumor measurements that were provided to us. Make sure your scans are collected on a specified time interval. So this is all, if you're looking at this, this is all sort of ambiguous, right? So we're going to measure some scans, we're going to collect some tumor dimensions, and then we're going to somehow make a call on whether the patient's disease got worse or not, right? So it's a little bit subjective. So back in 2009, we got the RESIST guidelines, right? It was published by a European group in 2009, and they collected tumor dimensions from thousands and thousands of patients and analyzed them to come up with rules to decide what a decrease or an increase in tumor size means, right? So they provide standard definitions for evaluation of tumor assessments, right? There are two groups of lesions or tumors, right? There's target lesions. These provide reproducible, repeated measurements. And then there's non-target lesions. These are other lesions, pathologic lymph nodes, or things that aren't so measurable, but are still of interest from a patient standpoint. So there are two categories of two broad definitions that they provide. One is response, like what happens if the tumor shrinks. That's an indication that the drug is working. And then there's disease progression. This is if the tumor grows, right? So if your tumors get bigger, if your patient's tumors are getting bigger, then this is probably not, the drug's probably not working. So response, there are two categories of response. One is a complete response, and this is all of your target lesions, pathologic lymph node, all your target lesions disappear, and your pathologic lymph nodes shrink to something less than 10 millimeters. The partial response is at least a 30% decrease in the sum of diameters of the target lesions, taking as a reference the baseline sum of diameters. Right, so it's a 30% decrease in the aggregate sum of diameters of the target lesions. We're going to call this a partial response. Disease progression is at least a 20% increase in the sum of diameters of the target lesions, taking as a reference the smallest sum on study. And there is an update to the RECIST criteria now that says that smallest difference has to be at least 5 millimeters or higher. Right? So this is a categorical outcome, right? If you're just for reference. Some other endpoints. So based on these response definitions, we can now classify patients as responders or non-responders, and with that, we can estimate a response rate, which is a binomial endpoint. So the objective response rate then is the proportion of patients with tumor size reduction of a predefined amount for a minimum time period. In this case, from the RECIST criteria, it's about a 30% decrease in tumor size. This includes partial complete responses. You'll see it PR plus CR. The significance of overall the objective response rate is assessed by its magnitude and its duration. So the primary endpoint might be the objective response rate, but having a response is meaningless if the duration of that response is short, right? And so what we want is overall, we want responses, we want tumors to shrink, but we want them to shrink and stay shrunk for a long period of time. And this duration for calling a complete or partial response, this duration estimate needs to be specified in the protocol. All right. Other endpoints based on tumor assessments, you can get event-free survival or disease-free survival. In event-free survival, we randomize before surgery or radiotherapy. Then the definition of event-free survival is the time from randomization to progression of disease or recurrence of disease that precludes surgery, local or distant recurrence, death for any cause. Disease-free survival, now we're randomizing patients after primary therapy, which is intended to be curative. So the patients are disease-free after they got this primary therapy done. Then the definition is time from randomization to disease recurrence or death from any cause. So multiple ways of thinking about time to either progression or death. Other toxicities in GOG 281 that you may or may not include in your study, toxicities assessed by the CTCAA criteria, which is now up to version 5. Self-reported acute quality of life measurements like the FACT-O or the FACT-O-TOI. GOG-NTX is a self-reported acute neurotoxicity measure, quality of life sorts of things, patient-reported outcomes. Exploratory analysis is where we get a tumor response rate in patients receiving trametinib after crossover. There were some others that I have not listed here. Right. So the first step in your design is to define your primary endpoint, and these are some options that you can choose. They're specifically related to cancer. If you're not studying cancer, then something else will do. The point is that you need to define them. So a quick review of statistical design. I'll take this one from GOJ 281. I got 2 slides here. This is a paragraph. This is a condensation of a paragraph that will occur very early in your stats section, and it has all the details in it that you need, but you probably want to add some more text around it. So this is the GOG 281 statistical design. We state that the null and alternative hypotheses with regard to the relative PFS event rate for trametinib versus the standard of care are this. So lambda T here is the progression-free survival hazard in the experimental arm. Lambda S here is the hazard for progression-free survival in the experimental arm, in the standard of care arm. So if this ratio is greater than 1, then my experimental arm is not working, and I'm going to accept or fail to reject my null hypothesis. And the alternative hypothesis is the same ratio, but if the hazard in the experimental arm is less than the hazard in the control arm, then this ratio will be less than 1, and I'm going to declare by some margin that the experimental arm is working. So lambda T and lambda S in this case, the notation are these are the PFS hazards in the experimental and standard of care arms respectively, and delta is the corresponding hazard ratio. I have a one-sided significance level of alpha of 0.025, power of 0.8 to detect a treatment effect size in terms of a hazard ratio of 0.5. This means I'm going to improve the median PFS from 8 months to 12 months, a 4-month improvement, or about a 50% improvement. At the final analysis, I'll do the final analysis when I have 213 events, right, out of a target of N 250 patients, right? So this is the basic design, and we're going to talk about some characteristics of that or how to think about that in the next couple of slides. So chapter 8 of the Friedman book from 2015 says that clinical trials should have sufficient statistical power to detect differences between groups considered to be of clinical importance. Therefore, calculation of a sample size with provision for adequate levels of significance and adequate levels of power is an essential part of planning. So the first phrase there, adequate levels of significance, we're talking about a type 1 error here, right? So if this is my hypothesis, where under the null, the treatment arm is not helping patients over the standard of care, then my ratio is greater than or equal to 1. Then the null hypothesis is true. Otherwise, we're going to call that it's false and we're going to reject. So alpha here, the type 1 error, is the probability of rejecting the null hypothesis when it is in fact true, right? So I want this probability to be low, right? So if in truth the treatment effect or the experimental arm is a dud, right, then I do not want to reject the null hypothesis because it is true. So that makes this a type 1. This makes an error of the type 1 variety, right? So this is the alpha this is the probability of a false positive result. It's the probability of incorrectly rejecting the null hypothesis when it is actually true. And this is the FDA's primary concern, right? So if you make this mistake, right, and reject the null hypothesis when it is in fact true, the FDA ends up releasing a truly ineffective therapy onto the market. And this is definitely not what they want to do. The other part of the FDA's concern here is that they have to deal with multiple studies, whereas we're only dealing with one at a time. Okay, so type 1 error is usually a very small number, right? Because we want a very low chance of making this kind of a mistake. The other thing we need is an adequate level of power. We want the type 2 error rate, sometimes called beta, to be small also. So here's my hypothesis. It's the same thing. Same hypothesis, no change here. Beta here, right, is the probability that when your null hypothesis is false, which means that the experimental arm truly is effective and you don't reject it, right? So it's the probability that you fail to reject the null hypothesis when it is in fact false. This is a beta or type 2 error. Power is the other side of that. It's just 1 minus beta. This is the probability that you do reject the null hypothesis when it is false. So this is power then is the probability under the null hypothesis or under the alternative hypothesis that your treatment is actually working, right? This is the probability of a true positive result, the probability of correctly concluding that the experimental treatment has an efficacy benefit when in fact it truly does. So the results of your experiment line up with the truth. This is good. Also, it's the probability of correctly rejecting the null hypothesis when it is actually false, is the other way of saying roughly the same thing. So in this framework, the decision, right, or the probabilities are conditional on whatever the truth is. So if the null hypothesis is true, right, if your experimental arm is truly a dud and you reject it, then you've committed, you have a type 1 error on your hand, a false positive. This happens with probability alpha. And this is some alpha that you're willing to accept, some chance that you're willing to accept. If the null hypothesis is true and you fail to reject it, right, this is correct, right, or you do not reject it. So your treatment is a dud. In truth, your experiment shows that your treatment is a dud, then you've got a correct answer. This is the true negative rate, and this probability is 1 minus alpha. On the other hand, if your null hypothesis is false, your treatment is actually effective, and your experiment rejects the null hypothesis, then you have a correct finding. This is a true positive result, and you want to have lots of power for this to happen. So you want the power number to be in the 0.8, 0.9 range, compared to an alpha number, which should be in a 0.05, 0.02 maybe range, right? On the other hand, if your null hypothesis is false, your experimental treatment is truly effective and you do not reject it, this is a type 2 error, right? A false negative. And you want this probability to be fairly low. And so these beta numbers are on the order of 0.2. But again, power here is just 1 minus beta. So this is these are the counterpoints to each of these points. All right. So there is an interplay, all else being equal, between the type 1 and type 2 decisions, or what should we call them? Calls or parameters, right? So importantly, the sampling distribution of your test statistic is almost always normal. So in statistics, what we do is calculate averages. Some of those averages get pretty fancy, but in the end, the central limit theorem is your friend. And by and large, if you have a large enough sample size, you can expect that the distribution of your test statistics will be normal under both the null and alternative hypothesis. So this is important because it makes a lot of the calculations for power and sample size easier, right? So in this block over here, this is the sampling distribution under the null hypothesis, right? So if my experimental arm is a dud, it's going to look like the treatment effect, or the sampling distribution of that estimate, is going to look like it belongs on the null hypothesis side, right? You get this, you determine what this is based on some existing evidence for your experimental arm, or for whatever the standard of care is, right? Okay, then over here, I have an alternative distribution, or under the alternative hypothesis, my test statistics it should look like this, right? It's going to be normal because of the central limit theorem. And then at some point, I need a rule, right? And depending on how I've defined these two distributions, and then I define my rule here, this, all else being equal, automatically defines what the type 1 error rate is under the null hypothesis, and the type 2 error rate under the alternative hypothesis. So the type 1 error rate is this red triangle down here in the lower right corner under the null distribution. The type 2 error is this sort of greenish triangle down here on the left side of the alternative distribution, right? So I can change all that. If these two distributions are fixed and I move this decision rule around, so if I move this decision rules, let's say over here a little bit to the left, then my type 1 error rate underneath the null hypothesis goes up, and my type 2 error rate under the alternative hypothesis gets smaller. And so I can adjust these things around. And this has to do with how you set your sample size parameters, and how you derive what those sample size parameters are supposed to be, right? So every test needs a rule to decide whether or not to reject that null hypothesis. You can have a p-value rule, something less than p less than 0.05. That's fine. Whatever rule you decide to do, the same logic holds. The other thing we can do is if we increase the sample size a little bit, then we tighten up the distributions under the null and the alternatives. These distributions become a little bit narrower, right? The tails are closer into the center than they are with a smaller sample size, right? And that changes the characteristics, the type 1 and type 2 characteristics also. So there are lots of ways to adjust these things. Is there any question about that? It's always confusing, I know.
I think we have about 5 minutes left.
Wow, we got 5 minutes. All right. All right. So here's your overall strategy for determining a sample size. You need to choose a primary endpoint, right? This determines which power equations you can use. You need to choose a significance level. If you're doing a phase 2 study, the alpha is probably in the 0.05 to 0.2 range. The phase 3 study, it's probably lower, about 0.01. You choose a power level. Power of 0.9 is very common, maybe down to 0.8. You choose the test and you solve for n, right? And I have several options here. This is about how the sample size gets collected or gets calculated. Don't do these yourself, right? You can use PASS or some other free software that will help you with that. There's same thing idea for a time to event endpoint, right? These are the quantiles. This is based on your alpha number, your beta number, whatever you've chosen, the effect size here, and the proportion of patients randomized to each arm. A little bit different way of doing it. So the minimum takeaway is you have to specify your endpoint. You have to specify your null and alternative hypothesis. You have to specify your effect size, which is the magnitude of the treatment effect that you're going to detect. You have to specify the statistical test. These are very common tests in a randomized study. It's very easy to do these analyses at the end. A significance level, a power number, and a sample size, right? For time to event, the endpoint is in, or the power is in the event count, not in the number of patients, just to keep in mind. So I have a bunch of slides here on interim analyses, which I will skip. And I will thank you for your attention. And please talk to your statistician early and often. They can't help you after the horse is out of the barn, right? So, any questions for me?